Greetings from Michigan State University, and welcome to EAB University's 2017 Fall Webinar Series, which is funded by the USDA Forest Service. This is Robin Usborne, and along with my EAB University colleagues, Cliff Sadoff and Sarah Stack from Purdue University and Amy Stone from the Ohio State University Extension, we welcome you to today's webinar, which is entitled Thousand Cankers Disease Threatening the Nation's Walnut Trees, and is being presented by Matthew Ginzel from Purdue University. Matt is an associate professor in the departments of entomology and forestry and natural resources at Purdue University, where he is also co-director of the Hardwood Tree Improvement and Regeneration Center. He earned his MS and PhD at the University of Illinois at Urbana-Champaign, and then went on to spend 2.5 years as a postdoctoral research fellow at the University of Nevada before coming to Purdue in 2006. Matt's research focuses on the ecology and management of native and invasive beetles that threaten the health and the productivity of our natural hardwood forests. Before we get started today, please know that we welcome your comments and questions, so feel free to type them in the chat feature, and we will be making notes of all the questions, and we will have Matt respond to them when his presentation is finished. After the presentation is over, I will be sending you an email with a short voluntary confidential survey that I'll hope you'll fill out and send to us so we can keep these free webinars coming. If you're one of the first 10 people to fill out the survey, we will be sending you an EAB goodie bag. For those of you wanting CEUs, if you would like a certificate indicating that you participated in today's live webinar, please complete the survey and send an email message to Amy Stone at stone.91@osu.edu. You'll also receive instructions in the email that I'll be sending out to you. Certificates will be mailed to you within a week of today's program. This webinar is being recorded and will be available for viewing soon at www.emerald-borer.info. You will also find the recordings for all previous EAB University webinars there. Thank you for attending today. And with that, Matt, please share your screen and we can get started. Looks good.
Okay, great. Thank you for the introduction, Robin. And it's my pleasure today to participate in the EAB University. Today I'll be talking about some of my ongoing research on Thousand Cankers Disease. And I want to point out from the beginning that much of this work is collaborative. One of my key collaborators is Dr. Jennifer Juswick, who's a plant pathologist, the U.S. Forest Service Northern Research Station in Minneapolis. And the work that we've been conducting on Thousand Cankers Disease is a good example of the collaborations of the two sort of sister disciplines of forest entomology and forest pathology. So I wanted to start off with a quick review of Thousand Cankers Disease. So it's the result of a complex interaction of 3 players. The first is the walnut twig beetle, Pityopterus juglandis. This is a very small beetle, a fungal pathogen, Geosmithia morbida, and a susceptible host, in this case, black walnut. So it takes these 3 players to form this pest complex and to cause mortality in walnut trees. So Thousand Cankers Disease originated in the Southwest and moved throughout the West quite rapidly, killing many black walnut in its path. It was then, at that time, there was a lot of concern around the idea that Thousand Cankers Disease might make it within the native range of black walnut here in the east. You can see the native range of black walnut in this figure outlined in green. So in 2010, it was first discovered in Tennessee, and in subsequent years, Thousand Cankers Disease has popped up in a number of neighboring states. In 2014, was the first find of the pathogen responsible for Thousand Cankers Disease, Geosmithia morbida, was found in Indiana in a plantation in Brown County. We have yet to find the beetle, walnut twig beetle in Indiana, and there are no symptomatic trees in the state at this time. So Thousand Cankers Disease has, its development of the disease is quite different in the east than in the west. So as I mentioned, in the west, that trees were killed quite rapidly. However, in the east, it appears that the disease progresses more slowly. In fact, some trees have been able to recover from the disease. In this photo here, this is work done by Gary Griffin at Virginia Tech. This shows the change in symptoms that were observed over 3 years in Knoxville, Tennessee, near that founder site where TCD was first discovered in the state in 2010. So you can see in 2010, this tree looks quite symptomatic. It has this thinning of the crown. There's significant branch dieback. There's flagging of the leaves that remain. This has about a 60% live crown rating. In September of 2010, there's moderate dieback. However, just 3 years later, this is the exact same tree. You can see that this tree looks quite healthy. It's recovered. There's been new stem growth. There's an abundance of new healthy foliage on that tree. The foliage just gradually increased over those 3 years until there's almost a full crown by the end of August 2013. And so Gary did some surveys of trees in Virginia and also in Tennessee. And what he found was that in Virginia, when in 2011 through 2012, when precipitation was low, that there was new TCD progression occurred. However, when precipitation was higher, it seemed as if that disease went into a quiescent state. And we saw the very same thing happen in Tennessee when there was high levels of annual precipitation. There was no new symptoms of TCD, and there was also increased growth and recovery of black walnut from the disease. So we started working in Ohio, in Butler County near Cincinnati. In 2013, there was an outbreak of Thousand Cankers Disease in that area. And you can see here that the Ohio Department of Agriculture went about a monitoring and survey program using pheromone-baited traps. And they found that there were set out 106 traps in 2013. There were 5 of those traps that were positive for TCD. And one of those traps had thousands of beetles in a cup. And that was in one residential neighborhood. And so that's where we started a lot of our work because of its proximity to Purdue. Jenny and I moved our research down to Ohio. But you can then see in subsequent years that they got zeros in their traps. So it seems as if that population there died out. So we set out to go into Ohio to understand the etiology or cause of this disease in the east. So you can see here are some symptomatic trees in Butler County. They have all sort of the classical symptoms of TCD, significant branch dieback, thinning of the crowns. And so we sampled some of these branches, first to find out the frequency of the cankers and also what insects are present on these TCD symptomatic trees. And what we found, what we were looking for are these classical Geosmithia-like cankers. So Thousand Cankers Disease gets its name because it takes literally thousands of cankers and symptomatic trees to kill a tree. So in response to the pathogen, the pathogen causes this dead area or a canker under the bark, and with high enough colonization or pressure from the beetle, these cankers eventually coalesce and affect tree health. So what we found was that in these 4 trees, that we found many Geosmithia-like cankers, these classic sort of look of a canker here associated with Geosmithia morbida. We found more of them in the branches than we did in the main stems, and we also found a number of buprestids and weevils and that are associated with some of these branches, and also a number of scolytine beetles that we'll be talking about later today. So then we looked at the frequency of isolation of 4 pathogenic fungi in Ohio. And what we found was that oftentimes we found Geosmithia morbida cankers alone. And so Jenny went back and assayed these cankers for the fungal pathogens, and also discovered that they had Fusarium solani and some Botrisphaeria, Botrisphaeria dithodia or Diplodia seriata as well. And oftentimes we found Geosmithia morbida associated with these fungi. So within a single canker, you found Geosmithia and also these other pathogenic fungi. So it shows that this perhaps is a complex of other canker-causing fungi that may contribute to the development of the disease. So what is currently known about these pathogenic fungi associated with TCD symptomatic trees? Well, Geosmithia morbida is a weak fungal pathogen. That's why it takes so much inoculum to kill a tree. One canker caused by Geosmithia morbida is not going to affect the tree very much. However, thousands, tens of thousands of cankers will eventually cause tree death. Botrisphaeria dithodia is a stress-related pathogen of walnut. It's considered an endophyte. And then the Botrisphaeria obtusa or Diplodia seriata, those are anamorphs of each other, is a pathogen of several woody hosts. Its pathogenicity on walnut is unknown at this time. And then Fusarium solani is a species complex that causes cankers on walnut, particularly following abiotic injury. And so from our first survey, we tested the hypothesis that, or developed the hypothesis that at least 2 other canker-causing fungi contribute to TCD symptom development in black walnut in the eastern US. So some of our guiding questions of this research is, 1, in the absence of walnut twig beetle, can multiple cankers caused by Geosmithia morbida lead to branch death? So if we were to inoculate branches with Geosmithia alone, is that enough to kill it, to cause death in a branch? And also, what is the relative virulence of Geosmithia morbida compared to these other putative canker pathogens of black walnut within its native range? So to answer these questions, we did some inoculation studies. And our thinking was that TCD may be more typical of a decline disease in the eastern US rather than a clearly defined fungal canker disease that would kill trees. So we did this work in 2 locations. One was in urban parks in Ohio, and also one in a black walnut plantation in Indiana, that very plantation I was talking about earlier, where we found the pathogen responsible for TCD. In each case, we inoculated these branches with local strains of Geosmithia morbida that Jenny had cultured. So to do this work was quite labor intensive, and for those who aren't afraid of heights, so we used boom lifts to get into this plantation. As you can see, this is a photo of that plantation in southern Indiana. It's a plantation that had been essentially abandoned, and so the trees are, there's a lot of invasive species in the understory, and the trees have not been particularly, or haven't been managed. There's been no real active management in that plantation for years. And so we did our inoculation protocol was to drill holes into the bark and to inoculate them with a slurry of Geosmithia morbida, and then cover that and wait to see the size of those necrotic or cankered areas, and also the resultant the health of that tree, or that branch rather, after following inoculation. So this shows you some of our methods associated with this. Early on we used these slurry with toothpicks. We've since then moved on to a different method, but nonetheless, this shows you some of that inoculation. And what we found was that the cankered areas with our sterile controls were quite small, but yet these two Geosmithia strains, 3 months rather, and 15 months following inoculation. And what we found was that 15 months following inoculation, that our cankers were actually were smaller, suggesting that this is an annual canker. The canker does not continue to grow in subsequent years. Also, that the cankers were in fact larger than those from our sterile controls. And so then we also extended this work to look at those 3 other, or 4 in one case, of pathogenic fungi. So this is the contribution of Fusarium and Botrisphaeria to the extent to which those fungi affect branch health. We did a similar protocol where we took branches from an individual tree and inoculated them, one branch each of one tree with one of the fungal pathogens. And so for this, we did a water suspension of the fungus propagules, or sterile water. We pipetted them into multiple drilled holes. The densities that we used were about 1 per 12 centimeters squared. And this is on the left here is a stylized locations of these 3 branch inoculations. And so we monitored these branches then 15 months later to see the extent to which they would affect tree health. And we measured that by branch dieback. And what we found was that with the sterile water, 8 of those branches, so this is 1 treatment per branch per tree, there were 8 of the 10 branches, there were 8 that were healthy. 1 showed 10% dieback, and 1 was in fact dead. With Diplodia seriata, there were a number, or 5 healthy, and then 1 each that showed the varying level of branch dieback. Interestingly, we didn't kill any branches with Geosmithia morbida. In fact, the most branch dieback that we saw from our inoculations with Geosmithia was only 20 to 30%. So it shows that Geosmithia morbida does not kill the branches alone. And in fact, fewer branches were dead with Geosmithia morbida than with many of the other fungal pathogens, and including our sterile control. This was in Indiana. We saw very similar results in Ohio, where we only used 3 of the fungal pathogens, not 4 in this case. And again, we saw with our sterile control, we ended up killing a branch, and we did not see any branch death resulting from our inoculations with the other fungal pathogens. So then we decided that we would look at the mean size of those cankers on Geosmithia, or on black walnut branches, in each of these locations 15 months following our inoculation. Here is a figure showing some of these results. Along the x-axis is the fungal pathogen, and also on the y-axis is the area of those cankers. And And what we found was that it was only Botrysphaeria that had larger cankers than any of the other treatments, and Geosmithia cankers were no larger than what we found with our sterile water control. Saw a very similar effect in, or we saw an effect in Ohio, the difference being that the Geosmithia cankers were somewhat larger than the sterile water controls, and the Thodia, we saw no increase over that of the sterile water. So our preliminary conclusions from this work is that in the absence of walnut twig beetle, none of the cankers coalesced on branches and trees. So we didn't see the cankers grow in subsequent years, and that Geosmithia morbida didn't result in branch death in our two sites. So Geosmithia morbida is a weak annual canker pathogen, and based on the size of the cankers produced. And we also saw a strong defensive response on the part of the trees. So in many of our inoculations, there was a lot of callus tissue surrounding those inoculation points. So the trees were able to wall off that infection. And other native canker fungi are equally, or in some cases, in Ohio, more virulent than GM was on black walnut. So our question is, would mass attack over multiple years of walnut twig beetle alone be sufficient to cause branch dieback? And some of our current work is directed toward answering some of those questions. So the part of what I want to talk about today was the threat of Thousand Cankers Disease in the East. And one of the questions that we are pondering and attempting to answer is that, is Thousand Cankers truly a disease, or is it primarily an insect problem? Meaning that because of the relative weakness of Geosmithia morbida, is it really only a matter of that it takes so much inoculum to get into a tree that's vectored by the beetle, that the disease in itself is really just a function of that pest pressure on the part of walnut twig beetle? And so from one of our first studies was a trap tree study, in which that was the study in which we found the pathogen in Indiana. And we started to rear out insects associated with these girdled walnut trees. And through the course of that work in Indiana, we found Geosmithia morbida on a weevil, and not on bark beetles. So our question was, what other ambrosia and bark beetles and bark colonizing weevils emerging from TCD symptomatic trees might be carrying the pathogen? So we went back to Ohio, excuse me, to answer this question. And this is in one of our cooperators' backyards. This is the very backyard where the Department of Agriculture, the Ohio Department of Agriculture, found thousands of beetles in a single pheromone-baited trap. And you can see this in this gentleman's backyard. There's a lot of mature walnuts. Also, there's some ash there. So the trees are mature, and so you can see that they're symptomatic. This tree, in fact, tree number 3, is almost dead from the disease. And so we sampled segments of these trees. He was kind enough to allow us to harvest these trees, and we took segments from 4 branches per tree, and then we took also 8 30-centimeter bolts of the stems of each tree, and we assayed those. We reared the beetles from them and assayed them for Geosmithia morbida. And we reared 8 species of beetles from the stem samples of TCD symptomatic trees. I'm just sharing the stem sample data with you today. And we found a number of ambrosia beetles, like Xylosandra crassiosculus, which is an invasive ambrosia beetle, and 26 of those. We reared 26 of those. We did rear out one Pediopterus juglandis, and we also reared out a number of weevils, including Stenomymus pallidus, which is the weevil on which we found Geosmithia morbida in Indiana. But 3 species, Xylosandrus crassiusculus, Xyloborina saxonii, and Stenomymus pallidus, accounted for 96% of the total number of beetles that we emerged from the stems of these symptomatic trees. So then when we assayed these beetles for the presence of Geosmithia morbida, 17 of those came up of the Xylosandrus crassiusculus came up positive, whereas only 15% or 15 specimens of Xyloborina were positive, and 13 of Stenomymus pallidus. So this was based on serial dilution plating of macerated beetle suspension, and also a fungal DNA extraction and detection from that same suspension. So we did it in two sort of parallel ways. This is work that Jenny and I have since published in Plant Disease. So what do we know about these beetles? Is that Pediopterus juglandis is an aggressive invader of walnut, whereas Xylosandrus crassiusculus attacks a wide variety of woody hosts, including walnut. It's a rather aggressive invader. It'll attack healthy trees as well as stressed trees. And Xyloborina saxonii attacks a wide range of woody hosts, including walnut. But little is known about Stenomymus pallidus, but what we think is that this is just a casual association it has with Geosmithia. For whatever reason, Geosmithia morbida was in those trees, or it picked up Geosmithia morbida in Indiana. And current questions that we're answering in the lab are the extent to which these beetles are able to leave with Geosmithia morbida on their bodies. Do they come to the trees? Are they able to inoculate a tree? Does a canker result from from this colonization by beetles that are carrying Geosmithia morbida? And the role of these other carriers of Geosmithia morbida in TCD symptom progression and pathogen requires further investigation. So it appears from a lot of our work now that this is the progression of TCD is stress related, that Geosmithia morbida alone is a rather weak pathogen, and it takes many inoculations from a walnut twig beetle to affect tree health. These other beetles are capable of vectoring, or at least they're leaving trees with Geosmithia morbida on them. But the extent to which they would kill trees or be able to cause the spread of the disease is unknown. Again, this is there are few Stenomymus pallidus that would attack a tree. In our trap tree survey, on 15 sites, we only reared about 400, or a little over 400 beetles, of which there was just a handful of those were positive. So this would be, if it's one beetle leaving and causing one canker, that would not affect tree health. So I want to talk a little bit today also about some ongoing work around the management of TCD in plantations. So some management considerations. And so I wanted to spend some time talking about work I've been doing out in Walla Walla, Washington. This is in southeastern Washington. These are walnuts that are being grown there as part of our ongoing work in the Hardwood Tree Improvement and Regeneration Center through one of our cooperators there. And there's an extensive outbreak of TCD in this plantation. These trees in particular were planted in the 80s, grow quite rapidly there. They're irrigated. And you can see this disease trees here with these thinning crowns and some branch dieback. This gives you a photo of one of this is the northern edge of that plantation, so much more of the trees are planted here. And so our cooperators began to see these trees declining. And Jenny Juswick was first to go out to the site and meet with the cooperator. And once she got there, she soon realized that this was a full TCD outbreak. Saw symptoms of branch dieback. Many of these trees looked like very symptomatic for the disease. And so that's when I came on the scene. We started to monitoring these trees using pheromone-baited traps. This is a common monitoring protocol for walnut twig beetle. This is the pheromone here, and it's impregnated into this bubble on a Lindgren funnel trap, and we just hang them from conduit. So I wanted to show you some of the monitoring that we found here. This is the site. These are part of the, these are walnuts. Some of them are genotype, they're of known genotype, and some were planted in 2001 as part of a provenance trial. That's this block here. And in 2009, we planted a series of grafted clones, and we're testing those against some select black walnuts within these plots. So we put up monitoring spot traps in each of these places. The BNT2 here is a place of where we have planted a series of pure butternut seedlings that were planted in 2007. It's interesting to note that these butternuts are also symptomatic for TCD. And through these monitoring efforts in 2016, we collected about 130 beetles per week in these monitoring traps. We did see some symptomatic trees within this 2009, what we call the Mickler plot, but otherwise, few of the trees were symptomatic outside of these large trees that I showed you photos of earlier that were planted in the 80s. What's interesting is that we confirmed walnut twig beetle, even though the trees weren't symptomatic. We peeled back the bark and were able to find beetles in many of these places. This shows you a picture of some of these trees growing in that plantation. Some of this branch dieback here is just the fact that they're growing in plantation, and so they're shaded out. We have 3 sites there. The other one is what we call the Yellow Hawk site. And this plantation consists of about 380 walnuts from our breeding program. There are symptomatic trees at the sites. There are beetles present, and we also collected about 34 beetles were collected at each of these per week. Just to the north of that that's another plantation, or that is that plantation, and just to the south of that is this provenance test of Indiana, Tennessee, and Oregon plantings. They were put in 2002, so they're quite a bit larger than that. And many of those trees were symptomatic. In fact, this shows you a photo of that. You can see this tree is dead from TCD. You see epicormic branching. This is one of my colleagues, Jim McKenna, who is the tree improvement specialist here at Purdue. He's a Forest Service employee. And as these trees were failing, the cooperator was stacking much of this wood as firewood. And so this became a real source for walnut twig beetle. And so our first recommendation to him was to remove this from the site. And then you can see that stacked firewood here. So this is with regard to managing the disease, this would be one of the first steps one could take to lessen the population of walnut twig beetle in that site. The third site is at Russell Creek. These are trees were put in 2009. TCD symptoms were most advanced in these big old walnut trees here. they were healthy in 2012. They started to show symptoms in 2014. There's twice as many trees that showed symptoms in the crowns of this planting here compared to the cottonwood or yellowwood hawk sites, likely because of this source population for beetles within these old trees. On this site, we collected an average of just over 1,400 beetles per week per trap. When I first got there in August of 2016, I put up some traps just to see the extent to which this disease, the severity, how severe this disease was. This shows the trap capture after just one day. And this shows the number of beetles that we captured in a week at that site. And so this was one of my students set out to start to count these, but I encouraged her to stop doing that. Perhaps we could find other ways of estimating population size there, other than counting all of these individual beetles. This is a nice photo that shows you the size of these beetles. They're quite tiny. And so we decided that we would try some management protocols within each of these plantations. And because there's 3 distinct plantations, we can test the efficacy of different management strategies. And so our idea was that we would, at that cottonwood site, the first site that I showed you, we would find these symptomatic trees, which are in red here, and treat them in the spring with bifenthrin. And so that would give us some knockdown from colonizing beetles, and then girdle that tree before the fall flight period, and then use that as a trap tree. And so that tree would then be removed in the during the dormant season and burned or discarded. And then asymptomatic trees would be treated with bifenthrin surrounding that in the fall. And then these untreated trees, asymptomatic trees, would be untreated. And within this planting, we identified our best trees, which we call our crop trees. Those are the ones that these plantations are in need of thinning. And so we would then thin some of those trees, leverage that thinning activity around some of these trap trees, so use trees that we would have thinned otherwise, and identify those trees that we would want to keep, and try to protect those with emamectin benzoate and propiconazole, so a fungicide. And this is work that's been done in collaboration with Don Grossman and at Arborjet. So we in the spring of this year treated a number of these trees with emamectin benzoate and propiconazole. We're also doing a test to see the extent to which propiconazole helps in protecting trees over Geosmithia morbida alone. At our yellow hawk site, where there's less pest pressure in that smaller planting, we would treat only crop trees with emamectin benzoate and propiconazole. So that would allow us to compare this insecticide and fungicide treatment to the trap tree treatment. And then in the Russell Creek site, where there's heavy pest pressure, we decided to test just the trap tree tactic alone. So in that case, we would those trees would be girdled following the fall flight period. There's 2 flight periods of Geosmithia morbida, or of the walnut twig beetle, rather. Sorry. And so it appears that fall flight is a stronger flight period, and so we would then target that fall flight period for us with our trap tree method. I want to show you these trees here. This shows you all of the cankers within that Russell Creek site where there's heavy pest pressure. This is where we're going to use trap tree alone. This is in the dormant season. Peel back the bark and you can see each of these cankers starting to grow. So an individual canker grows around an individual colonization spot from the beetle, and you can see with enough pressure that these would coalesce. So it really is death by 1,000 knife pricks with regard to TCD. I also want to talk about some current work. This is work that I've done in collaboration with Bill Klingeman and Janita Habdiabjic at the University of Tennessee, looking at volatiles of trees and the extent to which host plant volatiles might increase the efficacy of the pheromone lure. Many bark beetles are attracted to ethanol, which is a general stress response in the parts of trees. And so we wanted to test that and also understand whether we can manage some of these trees, manage some of these beetles behaviorally. So can we use attractants and repellents to move these beetles? There's a common repellent is of bark beetles is limonene. It smells like citrus. And we wanted to see whether the extent to which ethanol would increase the response of the beetle. So this was work that was done in Tennessee, in Blount and Knox County in 2013. We had 3 sites, and we used baited traps to test the extent to which the beetles were attracted to ethanol alone, and or repelled by limonene. We used the walnut twig beetle alone, and then the combination of the walnut twig beetle with ethanol and the walnut twig beetle with limonene. And these are the release rates of each of those. So you can see in this photo here, some of these traps were placed on transects along near symptomatic trees. Some of our early work, we tried, excuse me, hanging these traps from trees, but instead, for this work, we used them on transect. So here are the results. These are the treatments along the x-axis, and this is the mean number of walnut twig beetles per trap per collection period. And we found no attraction to the blank. Surprisingly, these beetles are not attracted to ethanol, nor are they attracted to limonene. In fact, when we pair limonene with the walnut twig beetle lure, we see a decrease in the capture over the walnut twig beetle lure alone, suggesting that limonene is a repellent for these beetles. And so in some areas, like in plantations that of high-value trees, it's possible, and something we are working on now, looking at the operational capacity of limonene to in a push-pull strategy where pheromone lures that the beetles are attracted to can pull beetles out of these high-value plantations. So these traps would be placed on the outside of the plantation, while inside the plantation we would use limonene. Or on those high-value crop trees, we could protect those alone, perhaps, with a limonene lure as a staple to the tree or nearby the tree. Also, interestingly, we saw no boost to the walnut twig beetle lure when paired with ethanol. Surprising in the fact that many beetles, as I mentioned, are attracted to ethanol, but not so surprising when you see that the beetles aren't attracted to ethanol. So I wanted to end today to talk about some management considerations. So in the East, it appears that Thousand Cankers Disease is not as severe as it is in the West. But I encourage landowners and municipalities and forest managers to monitor the health of walnuts. And importantly, do not harvest walnut in fear of a massive die-off caused by TCD. From all of our work to date, it appears that there will not be a massive die-off in TCD. And in fact, TCD symptoms are worse during drought stress. And those trees, when they become stressed, they're less able to defend themselves from TCD. And also to rely on science-based advice when making these management decisions. It's best to conduct management practices that keep walnut healthy and vigorous. So you can see in this plantation in Indiana that had not happened. This is some of that understory that's covered by Woody invasives, also vine control and proper thinning are important considerations. If TCD is in an area, in your area, it's best to reduce ambrosia beetle pressure. So that would be reduce cleaning up windbreaks, harvesting when the beetles are dormant, processing logs soon after harvest. It's a common practice to do dormant season pruning and thinning, or it's not common, but we encourage folks, if TCD is in an area, in your area, don't thin by just girdling trees and allowing them to stand for years at a time. And don't create wounds during the flight period. And more importantly, don't move firewood. It's a common consideration in thinking about all of these invasive wood-boring insects. It's likely that many of the spread of Thousand Cankers Disease was made worse and hastened by the fact that the firewood and wood materials were moved about. One can buy a walnut burl on eBay and ship it, and that is something that should be discouraged. So I want to mention these collaborators, Jenny Juswick, Jim English, Sharon Reed, also Bill Klingeman and Janita Abdiajic. We've received a lot of help from Phil Marshall and Megan Abraham from the Indiana DNR, Vince Burkle, John Day from the Ohio Department of Ag, Metro Parks in Butler County allowed us to do a lot of that inoculation work within the park system there. I also want to acknowledge the work of the hard work of students in my lab, Bridget Blood, Tyler Stewart, Gary Frazier, and Matt Passion. Funding for this work has been made possible from USDA NIFA, Forest Service, Purdue University, Indiana Department of Natural Resources. Some of that work from the Forest Service has come through Forest Health Evaluation and Monitoring and Forest Health Protection. We appreciate their support. And with that, I'd be happy to take any questions.
Okay. I'm kind of bummed that you don't know exactly how many of those little, TCB were in that plate. The little.
Yeah.
But anyway,
You got close. She estimated it was somewhere around. Well, I can tell you throughout the course of that growing season, through our monitoring, we collected over 250,000 beetles.
Oh my gosh.
So in that plate, there was somewhere around 10,000.
Oh, goodness. Okay. All right. Well, back to the questions. Anyway, Cliff Sadoff says, given that 17 of 26 Asian ambrosia beetles carry geosmithia, would you expect to find TCD in wet sites favored by the ambrosia beetles?
Yeah, so it's a possibility, right? So the one thing, though, is that even though those ambrosia beetles are leaving a walnut, right, they have such a wide host range that they would have to leave a walnut and then colonize a walnut. And that might not be the case. So they'd likely just go to the nearest tree that was weakened, right? Yeah, the question is if on wet sites, if site has some fact play in it. And that's some of the work we're doing now, is understanding the extent to which site factors influence the development of the disease.
Okay. Then the next question is, what is the potential for periodic extended drought in Walla Walla and the Ohio, Indiana sites, as well as Colorado, where TCD was first found?
Yeah. So in Walla Walla, those trees are all irrigated. So there we did some monitoring of water potential in those trees, and they were all quite high. If the extended drought in the east, if there is an extended drought, it wouldn't be unreasonable to think that you might see more symptoms pop up. That's the work that Gary Griffin did. One of my graduate students now has monitored We did through the course of one of his project. He did some transect studies and monitoring the health of walnut trees, and some were symptomatic throughout the last 3 years. Because there has not been an extended drought in that area, many of those trees recovered. But yeah, the evidence appears that it's drought related.
Okay. Do cold temperatures also impact the survivability of the beetles?
Yes. So that's outside of my area of expertise, but there's been a lot of work done in Minnesota on cold hardiness, and it appears as if they aren't that terribly cold hardy. That'd probably be a good have someone. It's Rob Vanette's been doing that work, and it appears that they are that cold temperatures can kill them.
All right, very good. That seems to be all the questions I have right now from folks. If they're when I send out my email to the participants, I'll share Matt's email in case you think of something you'd like to ask him later. But I know everyone is probably got other events and things to go on. So I will say to Matt, thank you so much for all that information. I'm glad we've been able to, have you present some information and kind of catch up on what's going on with this, with the Thousand Cankers Disease and that kind of thing, because we do get questions. And everyone, thanks for participating.
I think that's an important point, is when you do get questions, it's important to let folks know the state of our understanding here in the East. Mm-hmm. And it is not, I think it's cause for us to be vigilant, but there's no cause for alarm at this point regarding TCD. Does that make sense?
Yes. To be aware. Yes.
Aware, but we're certainly not, this isn't like EAB.
Okay. That's good to know too.